Good morning, students. So, today we are going to see the importance of phytochemicals from plants. You may be knowing that what exactly is a phytochemical. Phyto stands for plant, and chemical is the chemical substance. So, phytochemical is playing a very vital role in plants. You may be asking why phytochemicals are there in plants because plants are synthesizing its own chemicals, that is, phytochemicals, for its own protective mechanism. Like you can say alkaloids, terpenoids, steroids, the different type of phenolic compounds. Say phenolic compounds in the sense when you are simply cutting the apple or the banana, you will be seeing a very dark or a black color deposition. This indicates the secretion of phenolic compounds, right? So plants are secreting or preparing its own phenolic substances or various other phytochemicals to protect the plants. So it is a protective mechanism, just like in human beings, various type of immunological compounds are prepared or produced whenever we are facing or exposed to different antigens in nature. Plants are also synthesizing or preparing its own chemicals. It is a protective mechanism. But we are exploiting all this type of chemicals or the substances for our own use in the preparation of nutraceuticals and various food adulterants. It will be useful. There are different type of phytochemicals, and all this phytochemical, each phytochemical is having its own role to play. Say carotenoids are important in the maintenance of our eyesight. That is, if any deficiency of carotenoid that leads to eye blindness. That is what we call the night blindness. Okay, that is why the very important major project that emerged, like Golden Rice project, the very important presence of this project is just to supplement or increase the production of carotenoids in rice varieties. Each compound is having its own value. That is why, with or without our knowledge, right from the beginning, during the Stone Age itself, people start eating different type of vegetables, fruits, and cereals. Each plant compound or the component is having or part is having its own different type of chemicals. There are different type of, that is to thousands and thousands of phytochemicals are available. Today, I am going to concentrate only 10 different phytochemicals. So we are going to see how to check the availability of this different type of phytochemicals in plants. Like it may be a green leafy vegetable or a plant nuts or the seeds or a fruit, whatever it may be, how to check whether the plant is having the different type of phytochemical. That is two important phytochemicals only we are going to see. So I am going to give a very nice demo, live demo on how to check the availability of all these phytochemicals. Let us see this testing or the screening one by one. Please listen. Okay, students, I just want to explain how to check whether the alkaloid is present in the plant extract. Okay, we are using the extract of a fruit selected for our study today. I have taken a few drops of the plant extract. With that, I'm going to just add the hydrochloric acid. Okay, the care should be taken. The addition of HCl must be there along the sides of the test tube. Okay, once when it is added, we have to carefully add the adjuvants reagents. Okay, let us see. We have to add few drops of the reagents reagent carefully we have to add and see. Yeah, this way we have to add. Once when we added this, you can see a color change, a brownish or slight orange color change. It is visible. This indicates that the plant extract contained the required amount of alkaloid in it. Always we have to note that the intensity of the coloration indicates that how best the phytochemicals are available in the respective extract. Okay, so this indicates that the extract is having enough amount of alkaloid. Let us see the next one. Yeah, let us see how to check the availability of terpenoids. Okay, when we want to check terpenoids, we have to add 1 ml of chloroform. Let us see. I'm just going to add 1 ml of chloroform. Yeah, we have to add it carefully. See, I have added chloroform with the plant extract. After this, we have to carefully add 1 ml of sulfuric acid. So once when we add sulfuric acid, care must be taken. Just like that, we are not supposed to use this. Carefully, we have to add 1 ml of sulfuric acid. I'm just taking this because this is a concentrated one. We have to carefully add this along the rim of the test tube. Okay, just I am adding. See, we can easily visualize, or it is clearly seen that at the rim of this, we can see a red color ring appearance. You can easily visualize. This indicates that the availability or the presence of the terpenoids. Okay, you can easily see a ring-like spot that has developed. That has been developed. It indicates the presence of terpenoids. Okay, so the next one is steroid. I just want to check steroid whether it is available or not. Here, in the earlier one, we have added chloroform. Along with that, we have added 1 ml of sulfuric acid. So here, we are going to add only sulfuric acid alone. Let us see the color change. See, I am adding the sulfuric acid along the side wall of the test tube. Carefully, we have to add because it is a concentrated one. See, the color is totally changed. It is a dark violet color or a dark blue color. So this indicates that our plant extract is having enough amount of steroids. Okay, so this is the test for steroid. Let us see the next test. Yeah, the next test is for phenolic compounds. As I told you earlier, phenolic compounds are rich in different type of plants, specifically the banana as well as apple. You could easily observe this. The color change of the fruit or the stem of the banana, that indicates the availability of abundant availability of this kind of phenolic substances. So here, just to check the phenolic substance, say I have already taken the plant extract in the test tube. I am going to add few drops of ferric chloride. This is 5% ferric chloride. I just want to add this. See, I'm just adding few drops of ferric chloride. See, initially there was a change in the coloration. That is, it was blue, slight blue in color. This indicates that enough phenolic substances available in the plant extract. Okay, it is slightly bluish in color. Okay, so this is for phenolic compound. Okay, yeah, let us see how to check the availability or the presence of flavonoids. As I told you earlier, this is the extract what we are going to see. With that, I am going to add just few drops of sodium hydroxide. That is the NaOH. Yeah. Now we can easily see the color change. Yeah, it becomes a dark yellowish in color. So this indicates that the compound, that is a flavonoid, that is abundantly available in the test sample, that what we have used for our present studies. So this is the test for checking the availability of flavonoid. Now let us see how to check the presence of tannins in our plant extract. So this is the plant extract that I've already taken. So with this, I am going to add 10% of the lead acetate. Okay, this is lead acetate. I'm just going to mix it well. Just I am going to add few drops of the lead acetate. So you could see a milky white deposit at the bottom. You could clearly see there is a milky or a curd like deposit that has formed. It indicates the presence of tannins. So this extract is rich in tannin also. So this is the test for checking the tannins. You can easily observe this change. Yeah. So we are going to check the availability of saponins in our plant sample. So actually, for testing the saponins, we need to add 3 ml of distilled water. Okay, already I've taken distilled water. This distilled water will be added with this plant extract. Then we have to vigorously agitate this. See, we can change a foamy nature of this extract. I have added only the water. When it is agitated, we see the cloudy nature or the foamy nature of this. This indicates the presence of saponins in our test sample. Okay, so this is the very easiest method of test for checking the saponins. Let us see the test for carbohydrate. Actually, this is the molist test. Here I am going to take the plant extract. So with this plant extract, we are going to add the alpha naphtha. That is the alcoholic alpha naphtha. I'm just going to add very little. That is a few drops of this only. Okay, just listen. Few drop of this alpha naphtha is added. Okay, now itself you could see a color change. With this, we have to add, after the addition of alcoholic alpha naphtha, we have to add carefully few drops of the concentrated sulfuric acid once again, because this is a concentrated sulfuric acid. Just going to add few drops along the side wall of the test tube. You could see a purple ring that is developed at the ring. See, at the interface, we are seeing a purple ring. This is the indication that this extract is rich in carbohydrate. So this is the typical molist test that you may be aware of this. Okay, we can easily see a ring that is formed in between. Okay, by twisting this, you can check easily check the availability of this. Okay. So this is the test for checking the carbohydrate. Yeah, this is the test for checking the availability of proteins in our sample. So, after taking the extract, it was diluted with distilled water. That is why it is somewhat milky in color. Now, I am going to add few drops of nitric acid. This is a concentrated one. We have to carefully handle all these things, and I just want to take a little of this. We have to carefully add the nitric acid along the side of the test tube like this. See, carefully it has to be released. See, at the bottom, you could easily see a color change. That is a yellowish color change is observed. Is it visible? Yeah. This is the indication that our sample is rich in proteins. Okay. So initially it was white, now it becomes yellowish. This is the indication that the sample is having enough quantity of proteins in our extract. Okay, let us see how to check the availability of glycosides in our sample. So this is the plant extract. This is the diluted one. With this, I am going to add a few drops of sodium nitroproside, that is SNP, followed by we are going to add few drops of NaOH. Okay, let us see the color change. Few drops of SNP, that is a nitroproside, will be added. With this, I am going to add sodium hydroxide. See, a red color change has happened. So this indicates that our sample is rich in glycosides. So this is the very nice test to check the availability of glycosides in plant samples. Okay. Okay, students. Till now we have seen the test for various phytochemicals that is available in the plant extract. So initially it was colorless. Now you could easily see this is the test for alkaloid. A very nice change has occurred, and this is for terpenoids. This is for steroids, and this is for phenolic compounds, and this is for flavonoids, and this is for tannins. Very clear white precipitate that you could see, and this is for the glycosides, and this is for saponin, and this is for carbohydrate, and the last one is meant for proteins. So these are the various changes that we have seen. These are the easily identifiable tests for various type of phytochemical that is available in the plants that we are using in our day-to-day life, whether it may be a fruit or a nuts, or it is a cereal, or it is a leaf, green leafy vegetables, or whatever it may be. For all these things, these are the normal standard tests that can be easily followed and identified. So with this, I just conclude my today's lecture, and in the next lecture we are going to see some other topic. Thank you.